So I presented new data about targeting CD70 to eliminate leukemia stem cells with a novel antibody, cusatuzumab. So the background is that CD27 signaling, which is the receptor of CD70, promotes leukemia cell expansion and differentiation, and CD70 has quite a unique expression pattern. It's only expressed on leukemia stem cells, but not on normal human stem cells. Therefore, it's a good target. And cusatuzumab is a CD70 specific antibody that blocks signaling with CD27 and therefore mediates differentiation and reduces proliferation. But on the other hand, it's an ADCC-enhanced antibody that directly kills leukemia stem cells. What we presented was some preclinical work, how the strategy was designed and how the antibody was tested in preclinical experiments, and then an update on the phase 1 trial, and actually the combination of cusatuzumab together with HMAs had quite astonishing response rates with a very low toxicity. So we think at this point, given the limited number of patients, it looks quite promising. It was a dose escalation part with 12 patients, and we just reported on these 12 patients, and it was an expansion period, an expansion phase with an additional 26 patients, but we do not have the mature data on these additional 26 patients. So the response rate we reported was on the phase 1, and this was a response rate of 100% with 8 patients having a CR, 2 patients CRI, and the remaining 2 a PR. So everybody in every risk class and at every dose level responded to cusatuzumab treatment. So the next steps are definitely to await the maturation of the expansion phase data, so to have then more insights about potential biomarkers. We will look at NK cell levels, since it's an ADCC-optimised antibody on soluble CD27, which is a biomarker. This is more to work out which patients respond in the ongoing trial, and then 2 additional trials are already recruiting or in the startup phase, a larger globally recruiting phase 2 trial that compares 10 milligram per kilogram cusatuzumab to 20, so to define the dose to put further. And another platform study is just in startup, where we look at additional or interesting combination partners for cusatuzumab, and we think one of the most interesting that we currently know is venetoclax together with cusatuzumab without cytotoxic drugs. So in the dose escalation phase, we didn't achieve maximal tolerated dose. From this side, we didn't have some alarming signals, and the treatment-related side effects that we observed were very comparable to what is known from azacitidine. So we didn't observe anything special, just 2 patients with infusion-related reactions at the lower dose level of the study. So we think that the safety profile is quite good of the monoclonal antibody.